Hey guys, welcome to Chapter 8 Vocab. Chapter 8 is all about genetics and a little bit of proteomics. So the genome is the entire collection of DNA in an organism. So your genome includes your nuclear DNA, but also your mitochondrial DNA. And a bacterial genome includes its chromosome plus its plasmid. So all the DNA collectively in an organism is its genome. Now, DNA is often not in one big piece. It's often separated into little pieces that we call chromosomes. And they're not really little pieces. They're huge, ginormous pieces. So bacteria tend to have a single circular chromosome right here, whereas eukaryotes tend to have several linear chromosomes in their nucleus. So an organism can have one chromosome or many chromosomes, but it's the main DNA. In a eukaryote, the chromosomes are found in the nucleus specifically. Now, the DNA is made up of A's, T's, C's, and G's, right, that code for proteins. So each individual coding unit is called a gene. So if we made an analogy, all right, and you were reading a book, all right, so all the words in the book would be like the genome of the book. And all of the chapters maybe would be like the chromosomes, and each word would be like a gene. Okay, so all of those genes, the different flavors of genes you have are known as your genotype, right? But just because you have a gene for something doesn't mean you express it, doesn't mean you actually have that trait, depending, because a lot of times you have multiple genes that contribute to a trait. So you may have, so for example, sickle cell anemia is a disease that if you have both traits for sickle cell anemia, you get the phenotype, the trait or the disease of having sickle cell. But if you have one dominant and one recessive, so one normal hemoglobin and one sickled hemoglobin, all right, the phenotype is a little bit different. You get a much weaker form of the disease, all right? And if you have no sickled genes and only normal hemoglobin, then your phenotype will be healthy. So your phenotype is sort of the physical expression of traits, the characteristics that an organism has, and the genes are the genetic code. The genotype is the genetic code. So of course DNA and RNA are made up of building blocks called nucleotides, which consist of a sugar, a phosphate, and a base, either A, T, C, or G for DNA, A, U, C, or G for RNA. So those building blocks of nucleic acids are called nucleotides. So when we are dividing our cells, or when bacteria are dividing our cells, they need to copy their DNA. That's sort of the most important feature. Make sure they copy all the instructions to go into the next cell. So there's a lot of enzymes that are involved in separating, unzipping, and copying the DNA. So the first important enzyme in DNA replication is topoisomerase. So remember, the DNA is a double helix and it's coiled. So the first step in order to unzip the DNA is you first have to uncoil it. So topoisomerase sort of spins the DNA and removes the tension of that super coil. Then you get helicase, which comes in and literally unzips the DNA. So it breaks the hydrogen bonds that hold those two anti-parallel strands together and separates it into two single strands. Then some other enzymes come in the meantime, but another important one is DNA polymerase. So DNA polymerase literally means an enzyme that makes polymers of DNA. So this is sort of the key workhorse in DNA replication. It's the one that's making that second strand, that copy of the DNA. So once you've replicated your DNA, you've got a healthy cell going on, all right? If you want to make proteins, if you want to convert those genes, express those genes into proteins that then cause traits, okay, you need to do two things. You need to do transcription and translation. So transcription is the process of going from DNA to RNA. So you are making an RNA copy of the DNA gene. And then translation is the second step of protein synthesis, where you use a ribosome to translate the RNA into a protein sequence. So it goes transcription, translation. They're in alphabetical order. Transcription is DNA to RNA. Translation is RNA to protein. All right, in transcription, that first step, the workhorse enzyme there is RNA polymerase. It's literally an enzyme that makes a polymer of RNA. So DNA polymerase copies DNA into DNA, and RNA polymerase copies the DNA into RNA, and that happens in transcription. Okay? So then once that mRNA transcript is made, it goes to a ribosome where it undergoes translation, where the ribosome reads the RNA and converts it to protein. So the units by which, or the code by which it is read, is in these triplet base units. So a codon is a set of 3 RNA bases that is read by the ribosome and then translated into an amino acid. So each of these codons, or 3-letter codes, codes for a specific amino acid. So codons are triplet letter codes that are part of the RNA, specifically the mRNA, the messenger RNA. Now these codons code for amino acids. So for example, what does this say? ACU, ACC, ACA, and ACG are 4 codons, but they all code for the amino acid threonine. So notice they have the same first 2 letters, but that 3rd letter changes. So there's 4 different codons, but that 3rd letter in the codon is kind of flexible. And we call that wobble. So the genetic code has a lot of flexibility, which is great actually because it allows for mutations that are not, that don't have deleterious effects. So if you get a mutation from ACU to ACC, it's not a big deal because it still codes for the same amino acid. So having that wobble or 3rd position of that codon sequence actually protects us from mutations, or from harmful mutations, I should say. So we talked about the lac operon very briefly in Chapter 7. It's one of the ways that organisms can regulate gene expression. So bacteria specifically have a lac operon, which is a sophisticated and efficient way for them to regulate the genes that they need turned on if there's lactose around and they need to do lactose digestion and fermentation. So there's a repressor that binds to the gene when there's no lactose around, and when lactose is present, the repressor gets removed and the gene gets translated and transcribed and translated, and all of those enzymes that are needed for lactose metabolism get made. So that unit, that regulatory unit in the DNA that's got like the repressor and the promoter and the genes for beta-lactamase and all of the enzymes, that whole thing is called the lac operon. So we can use actually a lot of these enzymes from DNA replication to do some recombinant DNA technology. So recombination, and I guess I'm jumping ahead of myself, so the word recombination just means to recombine DNA from different organisms. So we can do that artificially with DNA technology, but bacteria and viruses do it a lot naturally. So technically lysogenic conversion, when a bacteriophage leaves some DNA inside a bacteria, that's a type of recombination. You're recombining some DNA from a virus and a bacteria. During conjugation, all right, when 2 bacteria swap DNA, that's also a type of recombination. So whenever you have 2 microbes of different types that do some DNA swapping, that new combination of DNA in the organism is called recombination. Conjugation is one method of getting recombination. So that's when you have a bacterial cell with a pilus, and it attaches to and transfers DNA with another bacterial cell. So the pilus is a sort of, an optional appendage, okay? And only bacteria that contain a plasmid with the gene for the pili on it, or pilus on it, can make a pilus. So that plasmid with the pilus gene is called an F factor. F in this case for fertility, because it makes them fertile, because pilus is like a penis. So F factor or F plus bacteria are ones that can make a pilus, and they can transfer that plasmid and turn other bacteria into pilus-containing bacteria. So that could happen through conjugation, through that mating process. Another way that recombination can occur is that some bacteria can just pick up DNA from their surroundings. This is something we call transformation, where they literally just absorb it through like pores on their surface. So I try to remember this transformation. The F in transformation, I think of fishing. Think of it as like, and I'll show you an animation in lecture where they've gotten some microscopy footage, and it really looks like these bacteria just like send out a fishing rod and they've just fished for DNA in their liquidy surroundings. They can just take it in. So transformation is when they fish for DNA that it's just like floating around in their aqueous environment. All right. Transduction is recombination by virus infection. So when a virus, or I should say a phage, a bacteriophage, infects a bacteria and it introduces its DNA and genetic material, that's transduction. Another way that genes and DNA can be recombined is through transposons. So transposons are nicknamed jumping genes. They're genetic material that can literally pop themselves out of a chromosome and pop themselves in somewhere else. So they're genes that can move around in a chromosome, but they can also jump from a chromosome to a plasmid or a plasmid to a chromosome. So transposons also increase genetic recombination. So sometimes when DNA is being copied, replicated, mutations happen. DNA polymerase has a very good spell checking ability, but when your genome is billions of bases long, even when you make a mistake 1 in a million times, that still adds up. So mutations occur. A mutation is a change in the DNA sequence. A point mutation is a change in a single base pair. So ATG becomes ATT. One of those bases changes. There's other types of mutations where like whole chunks of DNA get deleted, but we're going to focus on point mutations. Because even though they're small, just one base change, that change can have huge effects downstream for the cell. So one of the things that can happen is a point mutation can be a missense mutation. A missense mutation causes a change in the amino acid. So that one base change changes the codon, which changes the amino acid. And since proteins are dependent on their shape, and their shape is dependent on their amino acids, changing just a single amino acid can be enough to change the entire protein and ruin it, essentially. And that's what happens in sickle cell anemia. It's a classic example. A single point mutation causes this entire disease because that one point mutation changes one amino acid, causes a missense mutation, and then that causes the protein to fold funny, and it causes it to clump, and then it makes your red blood cells funny looking. So a single mutation in the DNA can actually be a big deal. But sometimes it's not. A silent mutation is a point mutation where it changes one of the bases of the DNA, but it doesn't change the codon. So you still get the same amino acid. So although the DNA sequence has changed, the protein sequence has not. So it sort of goes unnoticed by the body. It's not, there's a change in the genotype, but there's no change in the phenotype with a silent mutation. Okay, so some of these enzymes that bacteria have, many of which actually we'll talk about, are part of bacterial sort of immune systems, but they are things that we have used in order to do recombination, genetic recombination in organisms in a lab. So one important family of enzymes are restriction endonucleases, and these are enzymes from bacteria, and they cut DNA. An endonuclease is something that cuts, and they cut them at specific sequences, and it turns out that these enzymes are part of bacterial immune systems. They're there to cut up, they recognize bacteriophage DNA and chop it up, but we can use it to cut DNA in its certain sequences in order to do some like copy and pasting cloning. All right, so if that's the cut from the cut and paste, the restriction enzymes do the cutting, and ligase is the enzyme that does the pasting. So ligase glues nucleotides together. We've talked about this one. Reverse transcriptase is not found in eukaryotic cells. It's specific to retroviruses. It can make RNA into DNA. And this is useful because when we want to clone a human gene into a bacteria, since humans have more complicated genes, we can't really cut our DNA and paste it into bacteria, but we can take our RNA, convert it to DNA, because our RNA is more simplified, and we can convert that into DNA and put it in the bacteria, and then they can read it. So reverse transcriptase has been really useful, especially for recombinant proteins that are made for medication purposes like insulin. Gel electrophoresis is a procedure in lab where we can separate DNA on a gel. So we can cut up the DNA, we can separate it on a gel, we can see the relative size of the DNA pieces. We can use it for DNA fingerprinting and DNA typing. Another really common lab procedure, I don't even know what word I want to use, is PCR, which stands for polymerase chain reaction. So polymerase is the enzyme, we're talking about DNA polymerase here, that is used to replicate DNA. So this is basically DNA replication in a tube, and you do multiple rounds of it. So you can start with one piece of DNA and make millions of copies of it in a matter of hours just by doing DNA replication in a chain reaction over and over and over and over and over again. So that's what PCR is, polymerase chain reaction. So all of those are different tools that we have to do recombinant DNA technology, where we recombine DNA from different organisms so that we can produce human insulin in bacterial cells, or so that we can get rice to produce vitamin A, or plants that produce pesticides, etc. And then lastly, the term vector. So in microbiology, the term vector has different meanings in different contexts, but it always kind of means something that transmits information. So in the context of genetic engineering, a vector is something that takes DNA and puts it into a cell. So a vector is usually a plasmid. We can usually get plasmids into cells through transformation, or a virus, and we can get DNA into cells using transduction with a virus. So viruses and plasmids are both examples of vectors. All right, and then there's a couple of chapters, we'll talk about vectors of disease, which are insects that transmit disease. That's all. Bye.